Welcome back. If you're looking to get up to speed quickly, you're in the right place. Today, we're digging into something pretty exciting in breast cancer research.
Yeah, there's this preclinical study coming out of the University of Illinois Urbana-Champaign. They've been working on a new drug candidate, and the early results are, well, they're turning heads.
Definitely turning heads. The drug is called ErSO-TFPy, and the big news, the reason we're talking about it, is this claim it wiped out breast cancer in mice with a single dose.
Just one dose. It's a striking finding at this stage.
It really is. So, okay, let's get into why this is potentially such a big deal. How is this different from, the standard ways we treat cancer now?
Right. So the conventional toolbox surgery, chemotherapy, radiation, hormone therapy, they're the mainstays, absolutely vital. But, I mean, they come with challenges.
Big challenges, rough side effects. Sometimes the cancer fights back, develops resistance. It's tough on patients.
Exactly. It can be a really grueling process.
Yeah.
So finding alternatives or improvements is always a huge goal.
Okay, so ErSO-TFPy, what's its angle? What makes it different?
Well, what's really interesting is how it works. It doesn't go for the direct kill like, say, chemo often does. Instead, it targets something called the unfolded protein response, the UPR.
UPR, unfolded protein response. What is that exactly?
Think of it like a quality control and stress response system inside cells. Cancer cells, because they grow so fast and chaotically, are under a lot of stress. They desperately need this UPR system working overtime just to survive.
Ah, okay, so they're more dependent on it than healthy cells.
Precisely. Especially ER plus breast cancers. That's estrogen receptor positive, a very common type. They seem particularly reliant on this UPR pathway.
So ErSO-TFPy messes with that pathway.
It does more than mess with it. It seems to push it into overdrive. It overwhelms the UPR system so much that the cancer cell basically self-destructs. It triggers apoptosis programmed cell death.
So it makes the cancer kill itself? That's quite a strategy.
It's a fascinating approach, yeah. Exploiting a vulnerability rather than just brute force attack.
And the mouse study showed this worked really well.
The published results were, well, quite dramatic. For smaller tumors, they reported complete elimination after that single injection. Even with larger, more established tumors, there was significant shrinkage.
Wow. And what about side effects in the mice? That's always the next question.
Crucially, the researchers reported no apparent toxicity at the effective dose. The mice seemed to tolerate it well.
Okay, single dose, eliminates tumors in mice, no obvious toxicity. That sounds almost too good. What's the catch? The big but.
The big but is always the translation to humans, always. Mice aren't people. What works in a lab animal model very often doesn't work the same way or safely in humans. It's a huge hurdle.
Right, the infamous valley of death in drug development. Many promising candidates don't make it through.
Sadly, yes. The path to human therapy is long, expensive, and frankly, most attempts fail along the way, so we need serious caution here.
Still, the fact it was published in a respected journal, ACS Central Science, that adds some weight, doesn't it? Peer review.
Absolutely. It means the methodology and data have been scrutinized by other experts in the field. It gives the findings credibility at this preclinical stage. It's a necessary step.
Okay, let's play devil's advocate for a second and imagine this does translate well. If this pans out in humans, what are we looking at?
Oh, I mean, if it works and is safe, it could be revolutionary for ER plus breast cancer. A single-dose treatment.
Yeah.
With potentially minimal side effects compared to current regimens, that would be a paradigm shift.
A huge leap forward, less suffering, maybe more effective for some.
And potentially the principle could apply to other cancers too. Any cancer that heavily relies on that UPR pathway might be susceptible to this kind of approach.
So the potential impact is massive, but we have to keep our feet on the ground.
Absolutely. Cautious optimism is the phrase. It's exciting science, really important progress, but it's very early days. No human trials have even started yet.
Got it. Experimental, but definitely a significant piece of research highlighting where the science is going.
Exactly. It underscores why pushing the boundaries of basic science is so vital in the fight against cancer.
Okay, so wrapping up this deep dive, we've looked at ErSO-TFPy, a drug candidate that, in mice, showed this remarkable ability to eliminate ER plus breast cancers with a single dose, seemingly by triggering cellular self-destruction through the UPR pathway, and importantly, without obvious toxicity in those models.
That captures it. It's a smart approach, hitting the cancer where it's vulnerable, exploiting its own biology against it. Very promising, but still a long road ahead.
It really makes you think, doesn't it? What if future cancer treatments focus less on carpet bombing and more on these kinds of, well, targeted manipulations? turning the cancer's own survival tools into weapons against it. What other cellular quirks or dependencies are just waiting to be exploited like this? Something to ponder.